Good morning all. I am Mayur Kade. In previous presentation, we discussed a general introduction to pharmacology. That is, what is pharmacology, what is pharmacokinetic, and what is pharmacodynamic. Extending to that section, today we will discuss what is drug-receptor interaction. A receptor are specific membrane proteins which are able to recognize and bind to a corresponding ligand molecule and become activated and transduce signal to next signaling molecule. Whereas ligand is a small molecule that binds specifically to a larger one. For example, a hormone is a ligand for its specific protein receptor. There are several factors that governing a drug action, but more specific are affinity and intrinsic activity. Affinity is a measure of the tightness that a drug binds to a receptor, whereas intrinsic activity is the measure of the ability of drug to show a pharmacological activity. A signaling molecule binds to a particular receptor present on the target cell. After the activation of receptor, it will activate intracellular molecules which are called as secondary messengers, and the secondary messengers either activate a tertiary messengers or directly shows a biological effect. A signaling molecule either an extracellular molecule or intracellular molecule. A intracellular molecule directly activates cyclic AMP, that is cyclic adenosine monophosphate, cyclic guanosine monophosphate, inositol 1,4,5 triphosphate, diacyl glycol, and calcium. Whereas extracellular molecules shows their effect through a membrane receptor or intracellular receptor. The membrane receptors are, or through a membrane receptor, the effect shown by protein and peptides, amino acids and their derivative, fatty acid and their derivative, whereas through a intracellular receptors, a steroid hormone shows the pharmacological effect. The binding of drug to a receptor is determined by the following forces: it is hydrogen bond, ionic bond, van der Waals forces, and covalent bond. Among these four bonds, ionic bond is more stronger. Next, a binding of drug to a receptor. This drug may be a agonist or antagonist. As we discussed in the previous presentation, a agonist may be full agonist, that is a complete agonist, or partial agonist. A complete agonist shows a complete pharmacological activity as it is having affinity as well as intrinsic activity, whereas partial agonist having affinity but not a complete intrinsic activity, thus it shows a small effect. There are four types of receptor, that is ion channel receptor, G protein coupled receptor, enzyme link receptor, and nuclear receptor. The ligand gated ion channel receptors, after a binding of ligand to a ion channel receptor, ion channel is either open or closed. As shown in this figure, when neurotransmitter binds to a ion channel receptor, ion channel gets open and it will leads to a influx of extracellular ion to inside a cell. Second one is a G protein coupled receptor, which is a 7 helical transmembrane receptor. This receptor having a 3 subunit alpha, beta, and gamma subunit. After binding of neurotransmitter, the alpha subunit is separated from beta and gamma subunit, and this separation of beta and gamma subunit will activate the receptor, and this activated receptor will activate a secondary messenger system. The secondary activation system includes PIP2, IP3, and DG, and ion channels, and this secondary messenger will either further activate a tertiary messenger or directly shows the pharmacological effect. The third type is enzymic receptor, which is also called as tyrosine kinase receptor, whereas fourth type includes a nuclear receptor. These nuclear receptors, that is the intracellular receptors, directly activate a process of transcription and translation, that is the protein synthesis, whereas this tyrosine kinase link enzyme link receptor leads to phosphorylation of tyrosine kinase, and which leads to activation of cell signaling. We will discuss this tyrosine kinase receptor, that or enzyme receptor and nuclear receptor, in the next presentation. Also, we will discuss different factors that modify the drug action in next lecture. Thank you.